I'm David Donahoe, the session chair for our session 3 on scientific advances powered by AI and new science enabled by AI. I'm not going to put up slides and talk. I'm guessing my speakers will rather claim whatever little time I would save by not doing that. Our first speaker, I want to remind all the speakers in this session, could you stand in front of the podium so that the microphones can properly get your voice? If you wander around, then certain things may not amplify very well. Our first speaker of the session is John Jumper, who's a director at Google DeepMind. And in addition to some rather staggering awards and honors, identifies as a molecular dynamics dissident. Perhaps he'll explain what that means. John.
Some of my best friends do molecular dynamics, though. So it's really an honor to be here, and it's really exciting to talk at this session. And I want to tell you a story about AlphaFold and accurate structure predictions, and I'll kind of tell a part of the story. I'll tell you about the methods, and John will talk later about the assessment. How do we really know they're right? But I want to go to the next slide. Okay. I want to kind of start with an assertion that, of course AI needs biology. It's some of the most exciting applications of AI, but I think that biology also needs AI. The biology is extraordinarily broad. I think it's kind of maybe shares with something like material science being this incredibly broad. There are many things we want to study. There's, in the case of biology, an enormous amount of accidental complexity that arrives from evolution. And we've learned how to probe biology often with extremely clever people working extremely hard on a very narrow slice of biology, studying one particular protein system or one particular pathway, and doing brilliant experiments that uncover some really interesting information, and then we need to go move to the next one. And of course, humans have more than 20,000 protein coding genes. This takes a long time, that kind of getting this coverage of the complexity. And on your left, you'll see this, for example, minimal cell, right? This is an artist's illustration, but it's a quite accurate one. You can see the kind of complexity of the cellular environment. There's so much going on. There's so many interactions that we're going to need AI to be able to study all of these. And they're all, and many are individually different and specific. And so there is so very much complexity that we have to generalize from the incredible work of experimentalists in order to understand these systems. Now, I agree this looks suspiciously similar to Pushmeats. But I'll give a quick introduction to proteins. Proteins are this incredible system evolved that you can think of it, if our DNA is the instruction manuals for what exactly, one of the major answers is proteins. And using a relatively simple scheme, the stars for biologists, of 3 DNA bases become 1 protein residue. And there's 20 types of protein residues, so it's actually a fairly simple system from a computer science point of view. It maps nicely to our computers. Sequences run to hundreds of amino acids, and really importantly, simply due to physics, these fold up into complex 3D structures. And this is done because biology uses a library of chemical patterns, the 20 standard amino acids, and assembles them into straightforward chains. It solves the problem of how do you build complex things by mostly building simple things that are just polypeptides, and then letting the complexity arise from the interaction of these. Now, the shape of proteins has been experimentally studied for a long time and is an extraordinarily difficult thing to do. So, and I'll talk about just X-ray crystallography in this case. First, you have to figure out how to convince this large, kind of unwieldy protein structure to make a regular crystal. We don't have very reliable, we don't derive crystallization conditions. We try many things and see if we get crystals. If you succeed at that task, which is incredibly hard, you earn the right to take it and use the byproducts of particle physics, synchrotron radiation, although they're now purpose-built for this, to get very high-intensity X-rays to diffract from your crystal, and then you can solve a fiendishly difficult mathematical problem in order to invert the intensities in order to produce a protein structure. This takes maybe 1-2 years, give or take, to get a single structure. They're extraordinarily valuable for understanding biology, so we have something like 200,000 structures solved and deposited in the protein data bank. But that's nowhere near what we've determined about protein sequences, that billions of protein sequences have been determined, and even maybe the human proteome only has about 1/4 of it with experimental structural coverage. So we've got probably one of the nicest problems in science for machine learning in the sense of we have a data set of 200,000 structures where each one represents a year of someone that you're going to call doctor at the end of it, right? And an extremely strong relationship, going back to, as Bushmeat said, Anfinsen, between the protein sequence and the protein structure, yet we can't resolve that function. We've also had a lot of foresight by the experimental community that for more than 50 years, they've accumulated these data into standard formats with very skilled curators, and you essentially cannot publish a paper announcing a protein structure without depositing it. So we have this incredible wealth of, in one place, you have a data resource that represents the collective efforts of the experimental structural biology community. And you can think of this as also covering the full breadth of what is studied in structural biology, at least by methods that get atomic coordinates. And we can use this to make a machine learning system. And the cheesy joke I like to tell is computational alchemy. We just want to transmute the base metal of protein sequence into the gold of protein structure. The one on the left costs maybe a few cents to determine. The one on the right costs maybe $100,000. You can see in dark blue our prediction, in green the experimental result. This was predicted blind. We didn't know the answer at the time we predicted it. So we can do really extremely well. I'll skip the normal things where I talk about some of the validation so John can talk about it, but we've also, being a computational system, once we have an ability to predict, we can do it at scale. So we've made predictions originally for 21 model organisms, including human, now out to 200 million predictions. And so you can see, for example, the experimental coverage, or the representation of the size of the experimental data in the tiny purple circle here, and the size of our predictions, and still predictions. Not as good as experiment, but in blue. It's useful. And this was released in partnership with the European Molecular Biology Laboratory. So we talked a lot about kind of data, and I talked about data and how important it was, but it's not the end of the story. That it wasn't just a case of we sat around, we waited for deep learning to get good, we clicked download on the PDB, we stuck it in the predictotron, and then we ended up with a really great model. We had to do a lot of work, and kind of, for example, standard transformers don't really solve this problem. And the way I like to think of it is deep learning gives us these incredibly powerful tools, and certainly no one has missed that now. And we have different ideas like convolutional filters, graph networks, attention, which is the building block of transformers. And these are incredibly generic, right? They exist in chatbots, they exist in image generators. They're so generic they know nothing about proteins, right? They're unadapted. So all of the understanding of proteins has to come from the data presented to it. And these models are incredible at extracting signals from data. But we only have 200,000. Each one is incredibly precious. Each one is enormous effort by members of the community. So we owe it to the community, in fact, to adapt our machine learning. And instead of relying entirely on data, we try and meet the problem halfway and adapt our machine learning to the protein problem. And we did this basically, kind of earlier systems, and even our AlphaFold 1 system, were physical, were kind of the protein insights were on the outside. We stuck protein data in, we asked for protein data out. In the middle was a standard computer vision network. For AlphaFold 2, we built these insights, we adapted the machine learning, and I probably won't have time to go into exactly how, but we adapted the machine learning into the structure of the network. We built special blocks for how we process, physical data, how we process evolutionary data about the protein. And this enabled us to be vastly, vastly more efficient. One external study compared, they trained AlphaFold models on smaller and smaller amounts of data until they got to, I think, 1% or 0.5% of the protein data bank, and found AlphaFold 2 on a 1% of the protein data bank to be as accurate as AlphaFold 1 on the whole thing. So we have a pretty good claim to be 100 times more data efficient because of how we adapted the machine learning, because of how we built the right machine learning primitives for this particular problem. The result and the cleverness of this is in the modules like, oh, you can't see my cursor, is in the modules like the evoformer and the structure module and some of the other things. I won't go into this given the time. But I wanted to talk about something else that I think is really, really important. Okay, I've built the model. We can say it's typically good on average, but no experimentalist really cares about average behavior. They have a problem. They have one problem, they want a structure, and they're going to go use that structure possibly to do weeks or months of experiments on it. So how can they be sure it's right on their problem? And the answer is normally, well, you need some sort of confidence measure. And I won't say explainability, I'm going to go in a completely different direction and talk about what I would call black box confidence. So when AlphaFold produces a structure, right, the sequence goes in, we run AlphaFold, we get a predicted structure, and we get this second thing that is called confidence or predicted error. And so we claim that this is some location by location, residue by residue, prediction of the error. Now, how do you get such a thing? Well, let me first tell you how we get structure. So when we're training AlphaFold, we use a sequence of known structure. And so we produce our prediction. We have the experimental structure. And then we just measure the error. And in fact, we minimize not exactly this error, but we minimize an error. And so that's how you train these neural networks. And you change the parameters until the error is lower. But then how do you get the predicted error? And we do, honestly, the simplest thing you can imagine. So we knew the error at training time. So now, this second thing where we predicted the error, we can measure the error of our predicted error. We can say, is it right about how wrong this is? And the reason you should believe this exists is that you've all taken a test, and afterwards someone asks, how did you do? And you said, well, I think I did really well on the first question, not so sure on the second, I did terribly on the third. And it's not because you know the answer to the third question, otherwise you would have written it down, but simply we often know more about how good we're doing than we know about the true answer. And this same kind of thing actually works exceptionally well in protein structure. So we can predict errors, and this has turned out to be really, really reliable and calibrated. So we do the dumbest possible thing. We just ask the model, how sure is it? In a certain calibrated way that we know well in training. When we go to use it, you can think of everything to the right of the line you need to know the answer to do. But everything to the left, right, that original predicted structure and predicted error, you can do that on something where you don't know the answer. And so this is in essence kind of our answer to how do you know when it's right? Well, we characterize really well its own prediction of its error. So we predict LDDT, so we call it PLDDT. And we build this and another and really try and align so that, and biologists have actually, I've been shocked, one of the really great surprises of AlphaFold being widely available is that biologists really do look at the confidence, they really do care. And they help each other as well, saying, you know, oh, well, I see this, but I see the confidence is very low, so I'm not sure I really believe it, or I see the confidence is medium, so maybe. And they take action, they choose their experiments based on the confidence. And actually one of the most heartening things, you know, a few weeks after this was publicly available, it started becoming a meme on Twitter. If it's blue, it's true, because we would color the most confident regions dark blue. At the same time, do we know how AlphaFold works? And the answer is partially. We know some of the intuitions we had. Like any good experimentalists, we had some intuitions about what might work, informed by our prior experience, informed by principles. And we tried them. In this case, modifications, the architecture, right? We developed kind of the science of protein ML, at least locally for our problem. But then afterwards, we wanted to say, can we actually understand something more about how it works? And so we trained a small additional unit for every layer of AlphaFold that would try and give the best possible prediction for that layer. And so we use this, it's kind of like you're taking the exam again, and then 37 minutes into your hour-long exam, the professor says, pencils down, and you write the fastest answer you can. You didn't know you were going to have to give an answer then. And so you can see AlphaFold's best guess of the structure. It ends up in the place on the left. And you can see how it evolves, and in fact, most of it is kind of fairly physical refinement of the structure over the course of the network in fairly small moves, and you have to be careful on how you interpret it. But you can also see in the bottom left corner This gold region resolves at the end. AlphaFold couldn't figure it out early in the network, but finally it realized it could stick it up against here and resolve its structure. And so we're starting, I think, we don't yet understand how it moves, say, from frame to frame, but we've got an idea of, in a sense, what it was thinking when it got there. We can go do larger proteins. I'll skip in the interest of time. But I think it's also important to say, how do biologists use this, or how do they confirm it? And sometimes the experiment immediately confirms it for a certain technical problem called molecular replacement. But more often, biologists didn't really care about the structure. I was talking to Frances Arnold at lunch, and she says she doesn't care about the structure. She's looking for activity, right? And many biologists don't. And so what they do instead is they use the structure to build hypotheses of where this is going to go. I think one really beautiful example out of the Jang lab at the Broad and MIT is they had this new injection system for proteins that they had found in bacteria. They wanted to change what it targets. They said they had tried dozens or maybe hundreds of experiments trying to change what it recognizes on the surface of a cell to decide to inject its cargo. But they were doing it basically from the sequence in blind and couldn't figure it out. AlphaFold 2 became available and they ran it, and they almost immediately realized, oh, it looks like this, and these feet are probably what recognizes it, so let's replace the feet. And they got a general system that's able to target as a delivery mechanism for biologics into the cell. You're kind of using these insights in order to do another experiment. So what's really confirmed is not exactly the structure, and certainly not every detail is right, but what it gives you the hypotheses you need to carry out the experiments that you test well. We're continuing to work on this. A few weeks ago, we released AlphaFold 3. This is joint work with Isomorphic Labs, and this is really trying to answer the question of, okay, proteins are great, but proteins don't act in isolation, and not even protein-protein interaction. How do they interact with drug molecules? How do they interact with our DNA, and seeing how far we can push it? And I think also a really interesting case study in how far can we push things on very limited data. And I think this is a case in which we are getting further than anyone's seen before. I think some problems were really useful for, some problems were not yet useful for. We're still learning, but we also see the confidence measures hold up and become even more important in these kind of systems. But a lot of progress is being made, and we can overall do a lot of things. What can't we do? And I think there's a couple answers to what we can't do. I mean, some we can't do are simply, in a sense, limited by we are training against the protein data bank. We're ultimately locked into this world of what do proteins look like, of their coordinates. So we can't answer questions necessarily about doing chemistry or really dynamical changings of bonds, at least not very well. We're also, I think, limited in how much we can get kind of the full dynamical motion, although there's been a lot of recent work that starts to push and kind of find enough understanding of physics within AlphaFold that they can get distributions. And then finally, I think we're limited because we predict this wonderful thing, protein structures. It really is a lot of fun to work with. But we ultimately want to answer questions about biology. We want to answer questions out to why is someone sick or healthy. And we can give these structures to experimentalists. They're very useful. They apply their own human intelligence to interpret and design experiments. But we're limited in our ability to make these predictions directly or to hook them into some larger system that tells us, okay, well, this is how you should understand biology or how you should change things. And with that, I want to say thank you.
So I will hold questions for the panel. So there are several heroes of mine on the speakers lists for this whole conference, but one of those particularly is John Molt, who was very helpful to me in my understanding of the crucial role that community challenges have played in the whole progress of the protein folding story, and also in the rest of the machine learning story that's been so revolutionary. Professor Molt is a molecular physicist who's at the University of Maryland, and he pioneered the CASP challenge as well as some of the other important efforts that have been made in his field.
Thanks, David. It's nice to be a hero, but I'm not quite sure. I actually am not going to talk a lot about AI, but I am going to talk about the relationship between the type of thing I try to do and AI, and how AI I see is going to play a bigger role as we go forwards. So I'm going to start with the sort of thing that John Jumper talked about, this protein folding problem. I'm told if I press this button, I'll get us. Oh, I do, yeah, look at that. Okay, so you've heard about this, defined, I think Pushwitz said this, defined by Amundsen in 1972. It's a very nice, precise, and attractive problem, and because of that, it attracted a lot of people to work on it. And you just heard sort of the end of the story, at least so far from John, but let me ask you a question. Why do you believe anything he said? If you could say, oh, come on, he's got a paper in Nature, it must be okay. But actually, as I'll show you, you can't believe papers in the literature. So what I'm about, supposedly, is giving you trust in what he says. And I'll explain what I mean by that a little. So John talked about where we are with the problem right now, and we've got this wonderful solution, deep learning, down the bottom. But these other things are methods which were tried in the intervening 50 years between Amundsen and what John talked about. And if you look at these, there's a whole variety of different sorts of approaches. I'm an expert on these. Most of them failed, and I've got papers on almost all of them. And the question is, what was going on here scientifically? Partly, it's that this was really good, solid science. I'm not ashamed of those papers on failed methods. There were hypotheses, we tested them, they didn't work. In the end, deep learning did work. But there was something else going on, and that is we were having trouble communicating with each other what we really had achieved and hadn't achieved. And let me show you what I mean by that. As you know, problem essentially solved in 2020. If we go back to 1990 or so, here are some snippets from papers at that time. The first one, claiming an accuracy of 2 angstroms, which is not quite where we are now, but nearly. So we're folding up a protein. 1990, 30 years before the problem was actually solved, this is in Science, it's already done. And there are other things here, and put one of mine in here just to show that I'm not just picking on my colleagues. We were all doing this together. So what's going on here is an interesting story. I think the primary thing it's to do with trying to do science in a virtual world. So the methods of science, wonderful, don't get me wrong, but they evolve for a particular sort of science. And new sorts of science need adaptation to be able to cope with this. And what I want to talk about is one form of adaptation. And essentially you could regard it as using new technologies to try to get out of these sort of problems. The technology which we've actually used is actually very simple, but in 1980s you couldn't have done it. You didn't have ways of exchanging data and communicating. So what is this? Our supposed solution to this is something called CASP. And what we're after with CASP, I started this with Christoph over on the left, and these are other organizers, is to get more rigor, to be sure you can trust the results of the experiments. And out of rigor, to get trust. Again, to be able to say, I believe what John Jumper told me is really true. And the way we do this is very simple in principle. As John told you, experimental structures of proteins are coming out all of the time. And so we take the gap between something's about to come out publicly and it coming out publicly and ask people who are interested in this problem to predict what the structures are, bring in some people from outside to compare and get, as far as we can, an objective assessment of where we are, what methods are working, and so on. So we've been doing this for a while. It gets complicated in practice, as you can imagine, but I'm not going to go there. And so what have we seen over the history of this problem? So on the bottom, so this is target difficulty, so some targets are easy, and so you're high on that left-hand scale because you don't have to try very hard. But the ones John was talking about are really the ones over here. And you can see back in 1994, when we first did this, this curve is basically in the random region on this scale of accuracy. So not 2 angstrom accuracy as in Science, but actually random. And Then the good news is, of course, that over time, with those different methods contributing, we moved up. So I'm not going to go through the details here. The first use of AlphaFold 1 and other methods like it in 2018, the red line, actually is a big leap from where we were before. So where's my pointer? Here. From 2016 to 2018, when the first deep learning methods came in, we see a big jump. And then 2020, AlphaFold 2, we've got this black line, which is not at 100, but you don't expect to be at 100 because you're comparing with experiments, and experiments also have their difficulties. So what we like to assert about this is we've been able to carefully track what's happening, what works, and we hope, even though it took 50 years, that it helps push the problem forward more efficiently than relying on just the literature. Okay, so what do you need to get this type of method to work? I don't want to go through this in detail, but first of all, science often does work well without this sort of rather troublesome business. So you need to have a problem that needs it. But then secondly, and that was the nice thing about the Amundsen problem, you need a very crisp definition of what the problem is and what would constitute a solution. And then number 4, you need a precise set of evidence criteria to say whether you've solved it. Now, of course, with what I just described, comparing with experiment gives you exactly that. But that doesn't apply, as we'll see, to all areas you'd like to address. I'll jump to number 5. People have to do this. People have to put themselves on the line publicly and be compared with all the other methods. And in CASP, it's typically about 100 groups taking part, essentially the field. And so you've got to ask, why do people do that when they're going to be exposed in some way? And I think there is a deep kind of wish for better methods in science, and that's why people would go ahead. So this has been used, this is computational biology more broadly than structure, by about 20 different organizers to do these sort of experiments. Sometimes works well, sometimes works less well. I want to tell you quickly about one other, which is also computational biology. This is something Stephen Brenner and I started in about 2009, and it's about trying to interpret variants in genomes. So we've got now, of course, many of us in this room have our genome sequenced. Scattered here and there are variants which tell you something about your phenotype, and particularly your health-related phenotypes. And obviously, that's a computational problem as well as all sorts of clinical data to try and work that out. And I want to tell you particularly about just the first one here, things you might like to do with it, and that's diagnosing rare disease. You've got this something like 3 million differences out of 6 million bases, typically each one of us has. You're exhibiting some strange, unusual phenotypes. What of those 3 million is causing the problem? Now, this is mostly machine learning methods, conventional machine learning methods, and we've been working on it. Well, this is the slide on the picture on the right here is a summary from about 10 years of CASP effort. And what you're seeing here is the score on the bottom here on the horizontal axis from one of the best methods, Ravel, for trying to call whether a particular variant is causing pathogenic symptoms. And on the vertical axis, the actual probability calibrating against data, or rather measuring in blind prediction whether or not that's true. And so the line here is a calibration line for this Ravel method against actual observation obtained by blind testing. Now, why do I mention this particularly? The clinicians in all of these areas have been, I think, rightly skeptical of our claims as a community to actually be able to deliver something useful. And because other people involved as well, but because of the CASP, the KG results, I think they've accepted that this is a valid way of working out what to do with computational methods. And it's now, well, in the States anyway, a lot of serious deliberations about using this in the clinic, using this calibration. So again, it's about establishing trust. I'm not going to run out of time. Oh, okay. Okay, so I want to move on from this, though. This, again, is machine learning of some sort, mostly. And it's like the deep learning methods essentially black boxes. So what you'd like to know, you may know this is the causative variant, but what's the biology? Could you do anything about it? And so we've gotten very interested in trying to use these kind of community approaches to get at that. So this is yet another of these with Lindley Darden and Jan Pedersen. It's called Critical Assessment of Disease Mechanism. And again, it's to get at this mechanism thing. The example here on the left, this is a small lipid transport protein. Most people have the version of the E3 version, and there's one amino acid difference. This cysteine 112 to arginine 112, so this is the string of amino acids that form the protein. If you have that substitution, then your chances of getting Alzheimer's are very much higher. So this curve on the right here, the black line, is the normal chances of not getting Alzheimer's as a function of age. And you can see it falls off, it gets worse as you get older. But now if you look at the last red line here, this is the probability if you've got 2 copies of the E4 variant. And so by the time you're 80, about a 90% chance of getting Alzheimer's. So why is that? What's the mechanism? How can this tiny change in the protein produce this effect? So, of course, this has been highly studied. There are actually 10,000 papers on ApoE. And so I'll go to the literature, I'll look it up, right? So you go to the literature and you find this. So it's not one of my favorite sorts of trees. There isn't 1, 2, or 3 mechanisms proposed. Each of these things on the right is a proposed mechanism. There are actually 28 on here, and it's not complete. So most of these are in some way misleading. But what way? What could we do about that? And so what we're trying to do is a community experiment to address this. Now, you remember my conditions for when this might work. You need a precise definition of the problem. So how do you precisely define what it is you're trying to find here? We've come up, this is mostly Lindley's work, with this representation of what a mechanism is. So you start on the far left there. This thing delays before it comes on. Here's the genetic variant. Then it affects the protein, causes a change in the protein, causes some protein to move from one place to another in the cell. The key thing here is that results in a more acidic form of a thing called an endosome, which is in cells and sort of directs proteins to go here, there, and everywhere. And then a lot of downstream consequences leading to this increased risk. Do you notice these are causal chains? This causes this, causes this. And each step is colored. What you'd like is they're all green. You can see 1 or 2 places are red, and this is orange is more common. There's some data or not. By the way, I should say this is one of the 28 on that plot. This is mechanism number 4F that's been proposed. So is 4F a good mechanism? Well, it's very interesting, but it's got an awful lot of orange. What do we do about that? How do we establish when we should believe this stuff or not? So this is one knowledge representation to get precision about what we think we know. And then there's a second one. If I press the right button, it'll surely go forwards. Fooled me. Here we go. The second one is how do you get those colors? And so what we've proposed is a structured analysis of the evidence. So this is filling the criterion of what evidence do you use. We can't do the nice comparison with the experiment because this is the experiment. And you can see we've got about 15 different criteria here. And one goes systematically through these to try and find out how strong. Should you color it green, orange, or red? Now this works, and we've started working with the relevant community. We've got a pilot with 6 groups for this problem, and they're actually very enthusiastic. They like it, but it doesn't scale. It's very, very slow, especially to get this sort of information. But if you think about it, I'm actually talking about things which are mostly already in the literature. So this is where the AI part comes in. If it's already in the literature, why can't we just automatically retrieve this and do the analysis of the evidence? And the answer, as you all know, is that the AI tools for doing this are still imperfect. Question, how imperfect? So we've started to look at this. This is very, very Mickey Mouse compared with most of the things you've heard about in this meeting so far. We take ChatGPT. We use very structured prompts, though, based on these 2 the mechanism description, the evidence description, and there's a set of tasks you'd like the machine to do. Here they are: finding relevant papers, extracting key results, getting individual mechanism components, and you can see down the middle is my assessment of current performance. And sometimes it's good, sometimes it's moderate, sometimes it feels like pulling teeth. Overall, ease of use is very low. But compared with humans, overall, it's probably already a bit better. And so this is where we're at. We're trying to use these tools for this purpose. And the clear challenge, and this has come up already, I think the first introduction we heard made this point. The question we're trying to address is, how do you fuse this kind of tool with working with the experts to get the most reliable and rapid and scalable result? So of course, just to be clear, I think it's obvious, this variant is one of probably hundreds or thousands of cases where you'd like to do this. And if you can do it properly, you can learn a lot of biology and get a lot of potential therapy. Okay, that's it. Thank you.
We rely on our iPhones to unlock using face recognition, as I just did, and we rely on face recognition at airports, as you probably did on the way to this conference and so on, but maybe you never thought about how that all happened. The person who's had a front row seat at the creation of this technology is Jonathan Phillips.
Thank you. So I would like to talk about how I've done evaluations and the impact on face recognition over the last 30 years. I started working in face recognition in 1993. At this point, something called eigenfaces was state of the art. It was PCA to do face recognition. And also, algorithm development was the province of research and academia. In many ways, this was the start or the dawn of statistical machine learning in pattern recognition and computer vision. We've continued doing this to today. This has gone through the start of the deep learning era. And when you go through these, I will mention these where they come about. So David mentioned that we now use it at the airport. So we've measured this. So let's just start. I'm trying to find since I have to stay in my box, I'm trying to find the clicker. Oh, there it is. And I think this is the most useful one. Start with a simple problem we're addressing when you use it, just the technology. Here you put two faces in, and the question you want to answer, are they the same person or not? And the algorithm, in some sense, says yes or no. But this is not very good for evaluating an algorithm. So what happens really is you put two faces in, or you put a collection of two faces, ask the same question, and the algorithm gives out the similarity score. This is the confidence or the measure of how similar two faces are. You set a threshold, and if you do this here, on one axis you have a false accept rate, verification rate. This is a classic receiver operator curve, and then you see here, you set a threshold of where you want your operating point to be. And this is how we started measuring face recognition performance. So also go back to this, just to be in the background, what are algorithms like? So this is learning a face representation about 1994, Confessions. It was part of my dissertation when I was working on it. The input here is 58 frontal images to train it. You had an ancient learning function. It doesn't matter what it is. I did this on a Spark station sitting on my desk. And out popped a representation here, and this is just what it looks like. It turns out it was a wavelet representation, but that's not important. So now we go forward to the next example. This is from 2018. What you're training here is a ResNet with 101 layers. You feed in the algorithm 3.7 million face images of 38,000 faces. You have DCNN, cost function, and you now run in a GPU cluster. You come throughout and you get here is a standard characterization of what the faces would look like. true representation here would be a vector, say, with 1,024 dimensions. So in a sense, the same thing here for testing still holds. It's just that now a lot more time and energy goes into training, and the capabilities increase accordingly. So now I'll talk about progress of recognition. And here, the US government has conducted regular independent evaluations over 30 years. Because we go back to the beginning and want to measure progress, these are frontal facial images in controlled lighting. Think of passport photos or mugshots. Probably importantly here is that we tested the algorithms on sequestered data. So that means when we showed up to test them, they had not seen the data before. And since 2002, prototype algorithms have been sent to NIST for testing. And at this point, we started testing them in operational imagery plus others. One advantage of the algorithms coming to us is that we can test them multiple times in the same data to performance get so low. Another very important factor of this, which I'll go into, is that because we test them, we have all the raw scores. So we can look across the algorithms to see what the state of the art is and answer certain key questions. Also doing this, we did have change in data sets. As you'll see, as performance got too low, we had to switch to new data sets. So we asked two questions during this time period. The first one for the first 17 years was verification. This is the one I first mentioned previously. But we then moved to doing searching. So we got from the FBI 1.6 million mugshots. We then tested it. We had multiple images of the same person. So there's one image per person in here. We would then put in a person we knew was in the gallery. Our metric was the rank 1. So to get a correct answer, you had to search all 1.6 million faces and report the top match. This is something a human could not be done. And when I started in 1993, this would be just a dream. So let's go over this. We'll take a bit of time, hopefully this pointer here. So on this axis here is the year of the evaluation. I should say all these results from published results here. And at first we did verification. So we picked a false accept rate of 1 in 1,000. So that means if I tried to impersonate John Jumper, I would succeed 1 out of 1,000 times. And here we look at the false reject rate. We're looking for error rates. So this is how often the algorithms say that's not Jonathan. And what we found is in 1993, when we first started, that error rate was 79%. As we move forward using verification all the way down to 2010, we did move at one point to data from the State Department. Here, the false rejection rate, error rate was 3 out of 1,000. So that means now when I went up to get my money in the ATM, do a passport control, assume that you could acquire my image, the error would occur very infrequently. At this point, because this number is so low, we had to switch to the mugshot database. And here we use 1 minus the rank 1 retrieval rate. So this is the error rate occurs if the top match is not the person I'm searching for. So here in 2010, this is 77 times out of 1,000. Not bad. But then we went down here to last year, 2023, the error rate was 8 out of 10,000. So just think about this. We do this search 1.6 million and 8 out of 10,000. This is a case every couple years, every 5 years I would do this. I've been involved in evaluation, and my comment to my colleagues at NIST when we did this was, what mistake did we make? And we go search the data, we look at it, and eventually it was the answers were correct. It's just the program, the technology was improving that fast. So what this happens when you rescale it, it turns out the error rate halves every 2 years. So I re-put this on a new scale here, and along here's the year of the evaluation. I scale the error rate because we changed that database in the middle, and you see this then fit with a line here going all the way down, and what you see is happening every 2 years. But what's more interesting here, look at the data and tell me when the deep convolutional revolution occurred. You can't tell. So I think is that as another chart I have is the error reduced slowly over time as we learned how to use it. And probably this reflects that when it went from academia into the companies. These are mostly prototype systems that they slowly learned how to do it. And they had also at one point optimized for doing these mugshot types or passport type questions. So one of the things is that this technique is being adopted by computer vision. And what we see here, I have two. One is, and these are the ones that come to mind, one is the Pascal visual object classification. Andrew Sisterman was part of this. And here they had images they do, and this was 20 classes they would try to identify. A successor to this was, now I won't go over it, was the ImageNet, where they have 1,000 classes to classify. And I think this is old news by now. This is what enabled AlexNet to come forward. So one of the things I've done is that every time you throw an evaluation in, you get a new performer you've never heard of, a new method, and they would be up there with the best performers out there. We see that this is one of the capabilities of having these open challenges, is that it can identify new technologies. With successes like this, we saw John Jumper's show with AlphaFold, it starts people rethinking how to do this. So this is one of the powers of having competitions that are open and run in different methods. So I'm now moving to something else. Like I said before, we had all the data from here, so we do analysis. And one of the questions that I've had is, when is what's now an AI discipline first detected bias? So I got an email from the Council of Economic Advisors to the President, and if you go all the way through, they had a small section on bias in AI. They deemed it very important. So you go down here, there's one paper they cite, Furl, Phillips, and O'Toole. This is a paper we did, and it was published. Go to the next one, in 2002. So you see the other two they do prominently. This was done over 15 years before them. So this gave us the strength and the capability to analyze the data to first detect it. So after FRV2 2006, I again continued to collaborate with Alice O'Toole at the University of Texas at Dallas, who's a psychologist. And I used to joke that I understand now why the other race effect was her favorite problem. What we did now is that the other race effect is that people are better at recognizing faces of their own race. The question would be, would this hold true for algorithms? In the 2006 evaluation, we had down here, we had pairs of faces that were from East Asia, Caucasian, and we'd ask them, are they same or different? We also had algorithms submitted from East Asia, and algorithms were submitted from Western countries. And we created fusion algorithms, and we repeated the standard human tests, and what we saw was an other race effect for algorithms. So this is interesting that we're now using properties of the human visual system to analyze and ask questions about algorithms themselves. And in many ways, even though the other one appeared first, this algorithm was one that started a series of papers that we studied bias. The interesting thing here, if you look closely, this paper was published in 2011. Did we wait 3 or 4 years before we studied this? No, we did it right away. It's just that when we submitted it at this time, journals did not want it. We had to go through like 5 journals. So this is an example of how what seems like an uninteresting problem when you first start out comes to a major problem later on. So the other thing we did is another societal question, is there are people that are facial forensics examiners. They spend hours and hours comparing faces. And they're that can go and testify in court. So somebody can go in and say, here's Jonathan, here's a picture on that video camera. Tell me, is it the same person or not, the same face? And lock him away for the rest of his life. So this is a comprehensive comparison. They write detailed reports. They prepare to testify in court, and they have extensive training, 2 to 4 years. So we did this study. In the study here is that we actually used human performance and algorithm data from our evaluations to select what was 20 pairs of faces. They have to do the job. We also compared them to algorithm performance, and these were ones that came off of competitions we got that were run by another government program. So here we compared examiners to the general public. Examiners were better than the general public. Then we said, let's compare it to an algorithm. This is in 2018. And what did we find? Oh, excuse me, let me go back one. Okay. And we found they were equal to the best algorithm. So in 2018, the best algorithms compared with the best humans we tested. So we went one thing further. We said, let's combine or fuse algorithms and machines. And what did we find? They were better than examiners, and they were better than the algorithm itself. So this is a clear winner. We optimized performance on examiners and algorithms. And I can briefly tell you that fusion was quite simple. We took the algorithms, the human scores on a 7-point scale. We took the algorithm scores and rescaled them to the human scores, and we just added. Simply that worked. The reason we did this is known that a lot of times in fusion, oh, excuse me, I went back. A lot of times in fusion that you want to do the simplest stuff, and we want to make sure that it was robust, that it wasn't single here, that it would generalize to the next case we want to test. And this paper appeared in 2018. The one interesting thing now is we're designing another test, and remember the errors halving every 2 years. So this is about almost an order of magnitude improvement. At NIST, we can run from the best. The best algorithms now get 100% on this test. So this opens up a question that needs to be studied is people want human oversight of these algorithm comparisons. But when the algorithm is so good, what does human oversight mean? We believe this could generalize to other cases, for example, medicine, where they have large impacts, but at the same time, you do want the oversight. So now we're looking at the face ecosystem. So I've talked about the core technology here, but as we all know, when you put AI out, you need to understand how it works. So I call this the face recognition ecosystem. You want performance analysis. It's just as important how you capture that face when they go up to the monitor. I remember one time people from the New York City police I was speaking to, and they were telling me they needed this algorithm. I told them success depended whether they could capture good faces. You want to mitigate bias. Is it affected? Combining humans and machines, which I had previously discussed. Computer-human interface, and also the privacy. So I noticed that whenever I went through, in the US, I went through checking, they read my face, they used my driver's license to compare my face, and they had a little notice. After the comparison's done, your face will be deleted. There was also an option when I was leaving the country to go forward, and I noticed it said if you're a US citizen, we will delete this within 12 hours. So this is just as important as considering societal issues when you deploy a system. So the conclusions is that in 30 years we've had solid improvement. The contribution analysis computed to bias, performance on real-world images, and accuracy of face experts, and the adoption now by biometric computer vision in other communities. Thank you.
Several times this morning, we've heard about the importance of causality or causal inference for understanding what networks are doing or might do. And we will have a very interesting presentation on this by Caroline Euler. She's a professor at MIT and also in the Broad Institute, where she's the director of the Wendy and Eric Schmidt Science Center.
Thank you very much, and I'm honored to be here. So I work at this intersection between the biomedical sciences and statistics and machine learning, and have been really fascinated by how biomedical problems can lead to new foundational questions in AI, and in particular in the space of causality, which is what I'm going to talk about here. Now, we've of course already seen a whole lot of talks now about the biomedical sciences and what actually, why there is this unique opportunity and also necessity to bring AI into helping us with biomedical discoveries. One is data, as we've heard, and the other is questions, as we've heard. So on the data side, of course, now we have very cheap and accurate DNA sequencing. We have molecular imaging that is routine, very large-scale experiments. We have single-cell technologies which allow us to profile all of the 20,000 genes in single cells in 1 million cells per experiment. And just to show you in terms of data set sizes, so this is just Broad Genomics platform, so just one lab last year generated 100 petabytes of data. And this is not even taking into account any imaging data, so this is really just sequencing data. So that's on the data side. And then, of course, also on the question side, we've already heard this, we want to move on from studying the fundamental components of life by purifying them biochemically to actually much more abstract questions of understanding the whole regulatory logic of cells, of like which gene up and down regulates which other gene, how are proteins interacting, all the way to understanding the rules that govern developmental patterning, for example. So those are much more abstract questions also that we want to go after. Now, even though we have this huge data revolution in many of the scientific areas, we all know that it's really not science that is still driving some of the, the main key drivers of the foundations and the theoretical developments in AI today. But, in applications like online advertising, recommender systems, etc., actually these systems these applications have been very, very successful by just directly optimizing prediction accuracy. But in the biomedical sciences and in many of the other natural sciences, what we are really after is something different, right? Because we have natural laws that we want to understand. The ultimate goal is often to understand these underlying mechanisms, these natural laws. And so here we need to go from what we standardly look at as prediction to really causality and understanding the mechanisms underlying it. And so because the end goal is different, I think that also requires us to develop new foundations in AI. And now the question is, of course, what lets us get to causality? And we've heard a really great talk this morning in a completely different field, right, in online in online education, where we can actually perform large-scale interventions. And of course, in the biological field, we also have this huge hammer, which is actually being able to perform large-scale perturbations through genetic or chemical tools. And so there is, if this is paired with the right AI tools, there is a unique opportunity to really, from these perturbations, be able to understand what the underlying regulatory logic is. Right? Because if you, for example, use CRISPR to, say, knock out a particular gene, then only things that are downstream will change. Right? So if you do a perturbation, only things that are downstream will change. So you may be able to understand what is upstream, what is downstream, and from that kind of try to reconstruct what this regulatory logic is. Now, the problem is that, in fact, our current AI tools, because they were developed based on these types of applications, are actually very, very bad at understanding, or just working with interventions themselves. And here's a very, very simple example. I mean, we all know, right, that if I choose a cold and gloomy day and I go out and I give out a lot of free ice cream in order to increase ice cream consumption, that obviously doesn't change the weather, right? Just because ice cream consumption is downstream causally of good and warm weather. However, if you, in this case, still DALL-E 2 or DALL-E 3, I'm sure, do similarly, if you ask it to generate an image where you're showing an ice cream stand where they're giving out free ice cream, it will actually always put good weather in the background. Because it's like very, very good at at the operation, which is conditioning. Okay, if I condition on a lot of, high ice cream consumption, well, that changes the probability of weather to being, a higher probability of good weather. Similarly, if you condition on good weather, right, that means that with a high probability I have high ice cream consumption. So it's very good at this symmetric operation. It's actually very, very bad. And that's just because it wasn't built to do the other types of reasoning, this interventional reasoning. It's very, very bad at trying to represent and figure out what is actually, because now I need to know what is upstream, what is downstream, right? So if I intervene on ice cream consumption, if I put that high by intervening on it, well, the weather doesn't change. But these are exactly the types of questions that are critical in the sciences, right? Because we want to understand in the end, we need to be able to predict the effect of unseen interventions. We've heard this morning, right, in Kim's talk, like the space of all drug-like molecules is 10 to the 60. We'll not be able to even generate them, and let alone like actually apply them and try to figure out what the effect is of all these different drug-like molecules and which ones will be the right ones to do. Same in the genetic space. We have all these 20,000 genes. If we're just thinking about even just, we can do all kinds of perturbation experiments, but say just knocking out different genes. We can knock out any of the 20,000 genes or any combination of these 20,000 genes in any of the different cell types. And so we'll never be able to do all these experiments. So we need to be able to predict what the effect of an unseen intervention is, like these types of, what happens if we increase ice cream consumption. And I think maybe it's a little bit different than in the online education system where, you often want to generalize, you have a set of interventions and you want to generalize when you've seen some of them, well, what will happen if I apply this intervention to another person? Whereas here we really, the hardest part is that we have to generalize across interventions that we have never seen, and we'll only be able to do very, very few experiments. So I want to, just also to relate to the talks that were now in this session, I chose to talk about this example about cancer immunotherapy because we ran an AI challenge in this space. And this is one setting where, the question comes up of like, well, which experiment should we run? And so we all know of, these amazing advances that we've seen in cancer immunotherapy in recent years and the unique opportunities where, our immune system is actually able to kill cancer cells. However, cancer cells are very good at evading the immune system. They send out signals that make our immune cells go into this terminal exhausted state. And so what is underlying cancer immunotherapy is that we would like to be able to somehow figure out how to perturb our immune system, in particular the T cells, so that they are, in fact, remaining in the activated or in the effector state. And so, the challenge of cancer immunotherapy is, of course, it works really, really well for some drugs, for some cancers, and for some people, but we would love it to work for more cancers and more people. So one way of studying it is to try to identify what should we target in these cells in order to actually make them stay in this effector state. And so one way of studying it is just by genetic perturbations. And so here is the data and the challenge that we ran. And now this is again a setting where, performing all experiments is just simply not possible right now. And will never be, because even if you just look at genetic perturbations on genes where you're knocking out different genes, we'll never be able to do all possible high order combinations of 20,000 genes. So here what we did is we provided 70 different knockout experiments. They have to be done in vivo so that you actually have it in a cancer context and get to see what do the T cells do and which cells do they go. So you have some perturbations where, and I'm also looking for, here we go. So you have some perturbations, right? You would like to have perturbations which are depleting this terminal exhausted state. So this here is a pretty good perturbation where you have very few cells that go, T cells that go into this terminal exhausted state. You would like them to actually remain over here. And so, now here we provided 70 different knockout experiments, and we're asking, well, out of all the 20,000 that we could have done, which ones should we have done that would have been better at moving the T cells into these states? And so I think here, these are, I think, one of the first competitions where we're actually asking AI researchers to define the next experiments we should be running. And then we are ranking the algorithms based on these. So we're actually running the experiments and then ranking algorithms based on these new experiments that we ran. And I think we have to move to this space where we have new AI science competitions, which are actually about, helping us define what are the experiments that we should actually be running. And also, hopefully, these types of AI competitions will also move the AI field more towards, these more causal prediction tasks, and not the more standard prediction tasks that we have seen so far. To put this into, a more formal language of what do I mean by a causal prediction task and how is it different than our standard prediction tasks, I really like this causal hierarchy here by Julia Pearl, where at the bottom, this is like what we standardly do in any of the prediction challenges, which is just based on association, asking, if I condition on high ice cream consumption, right, what is the probability of weather? Or if I condition on good weather, what is the probability of high ice cream consumption? Then in fact, here at the second level is what I have talked about in this talk, which is what I call causal prediction, which is the level where you're trying to answer questions, what will happen if I provide or, do the intervention where I'm providing a whole lot of ice cream, okay? And so that's the intervention. This is not a symmetric. Operation. Here, you actually need to know something about the underlying regulatory networks or the causal networks. And then all the way at the top is actually what most of causal research is nowadays about, is counterfactuals. But that to me, and so the types of questions here is, what would have happened had we all eaten a lot of ice cream yesterday? Okay? So that's in fact a much harder question than what will happen if we all eat a lot of ice cream in the next 5 minutes. Okay? And so a lot of the causal research is all the way at the top. A lot of the more standard machine learning research is all the way here at the bottom. But I think the level which is really critical for AI and science is actually this middle layer that is unfortunately not really getting as much attention as I think it should. Where we have questions about predicting the effect of unseen interventions. And even, in fact, the deforestation question that was brought up, it's not, I think, a counterfactual question. It's actually also in this level here. Because it is about predicting the effect of an unseen intervention, not having to do this in a whole other system where we're currently not in yet. And also in here falls all of the questions that we've seen, or what we've heard this morning also in Christine's talk, I think, where it was like, what we care about is the things where we can then do experiments to validate, right? So counterfactuals, you'll never be able to validate. For certain questions, that's what you care about. But I think in most of the biomedical questions and scientific questions in general, you are actually in the second level. And so we really need new AI theory that can actually help us in this second level, which I understand in Julia Pearl's hierarchy is only the Neanderthal level, but I think that is actually the really interesting level, and hopefully we can move a little bit more from associations actually to this level here. And if we can answer questions like predicting the effect of an unseen intervention, the types of questions that we are very excited about, and of course relates to a lot of the things that we've seen today already, is, well, then we should be able to identify the optimal intervention in order to move a cell to a particular cell state. Say, for example, in cancer immunotherapy or in reprogramming, rejuvenation, etc. Types of questions that I care about a lot. But again, if we would know the underlying causal system, well, then we would know what the optimal intervention is in order to move the system to a particular state, but we don't know it, right? So we need different rounds of experiments where we identify, where from interventions we learn something about the underlying causal system, which then allow us to actually produce better interventions. And maybe again, like slightly different than in the online education setting, and maybe also actually a lot of the protein setting, is that in this biological setting here, when we're thinking about cell state transitions, the experiments take long. So what we need is we can do a lot of experiments in parallel, but we only have, say, five rounds of experiments. And so we don't have this infinite horizon, etc. Like none of that is actually relevant for these types of experiments that we need here. And maybe I also, since there was some talk about multiple modalities, right? And maybe what is the power of that? And I want to bring up like how that actually also connects to causality, because that's often not talked a lot about, and it's something that we do a lot of research in. So in fact, interventions is one way of getting at causality, and we all talk about that and know this, right? But there is a much deeper kind of a deeper mechanism underlying it, why interventions help us get a causality. And there are many other types of things that actually help us or can help us get a causality. Interventions are just a way to introduce heterogeneity. And really what we're doing in order to get a causality is trying to identify what is invariant in the underlying system. So what is invariant to the particular intervention that we're doing that is like upstream instead of downstream? And there are other ways, of course, of adding heterogeneity or making use of heterogeneity, one being having access to multiple data modalities. So we work in the biological setting where we have, say, sequencing data and imaging data, or in the medical setting where we have ECGs and MRIs, etc. So in all of these settings where you have different views of a system, so different data modalities on the system, how you can actually use that in order to get a causality is to ask what is invariant to how you're looking at the system, right? What is truly causal should not depend on whether you're looking at the system from the left or from the right or from the top or from the bottom. And so you can use different modalities to look at what is invariant to how you're looking at the data, and through that actually filter out a whole lot of stuff that are associations that don't come from real causal relationships. So really the underlying logic of why interventions help you is actually, it's really just about generating heterogeneity, and you can generate heterogeneity in many other ways as well, and use that in order to get a causality. So you should really always make use of multimodality whenever you have access to it. And so I'm very excited to direct the Eric and Wendy Schmidt Center at the Broad, where we're really building this two-way street between AI and biology. And I hope I was able to convince you of something that I believe very strongly, that the biomedical sciences are uniquely suited not only to being one of the greatest beneficiaries of AI, but really also one of the greatest sources of inspiration for novel developments, foundational developments in AI. So thank you.
Of the panelists that I've chaired, the next speaker has the distinction of having written the most books that I personally own. I own one book of Jonathan Phillips, but I own two of Andrew Zisserman. He's a very well-known contributor in computer vision, and in fact, I've done a paper that a lot of the work involved using the VGGNet, which I believe that he originated with his co-authors. He's also a member of the Royal Society. Please.
Well, thank you. It's a very nice introduction. Thank you. So I'm going to talk about something slightly different to other people have talked about, which is how AI can be used to enable science. Okay, so to be clear what I mean is that we've seen wonderful examples here of how AI can solve what we could call grand challenges in science, one after another. And we also saw that the people acknowledged that this stood on the shoulders of others, building on work over multiple years. What I want to do in contrast or complement it is how AI can be used to help scientists do their own research. It's a different take. So this is a bit like Alex talked about data being AI ready. And what I want to talk about is how to make, or give examples of how to make data AI ready, how AI can be used for that. Okay. So I'm going to be talking particularly about computer vision, because that's my background here. And what we found in working with other disciplines over like 10 years is that often people, the scientists in that discipline, have massive amounts of data, thousands of hours of video, terabytes of data, but they can't use it. And the reason they can't use it is because it's not annotated. And the point of this talk is that we can use AI to annotate this data automatically. It's beyond anything they could do. And I'm going to illustrate this with 3 example projects that we've worked on over the last decade. One in animal behaviour, one in materials, and one in conservation. Okay, so it will become clearer as we go along. So the first example is animal behaviour, and this is for chimpanzee face recognition. So we're working here with some scientists in anthropology and zoology at Oxford. They've been collecting data for 14 years. They had hundreds of hours of video, and many students have collected this data, but they couldn't use it for their own research. And the first thing they wanted to be able to do is to label every video frame with the identities of the chimpanzees who were there. Okay, so here's a typical bit of video. You've got these chimpanzees walking around. So they want to know which chimpanzee is there. Why did they want to do this? Because what they were interested in were the interactions between the chimpanzees. They wanted to actually build a social network between the chimpanzees, but they couldn't do that. So we came in and we built a system to enable that. So it has a number of stages. We got the original videos. We built a face detector, because at that time there wasn't a good face detector for chimpanzees. We tracked these faces through the video. And then we built a network which could recognise the chimpanzees. It's very much like Jonathan had built or had established for human faces. We did a very similar thing for chimpanzee faces. Okay? So when we'd done all that and we'd processed these hundreds of hours of videos, then they could do the research they wanted to do, which was to analyse the data and get these social networks. Okay? I'll give you a visualisation of what this is. So you'll see a video sped up, save time here. So here's some chimpanzees in the wild. And then you'll see the face detection and tracking. Okay, so they're not identified yet. Okay, each track numbered. And now we identify them and get male or female as well. Okay, so this is, as I say, this has been done, and now they can do the research they want to do. But before that, they couldn't. And what happens usually in these engagements, we publish a paper with them about the method, and then they can publish papers on the thing they really want to do, which is analyse social networks. And then that leads on to other people in their discipline, in their field, building similar methods, and that's what happened. So I'll give you a couple of testaments, what a difference this made. This is the people we work with, and if you look at the bits in blue, the first one saying, it made it possible to address questions that were impossible to tackle until today. And the second one, this was a step change. It's a breathtaking. So these are very happy scientists now, very, very good scientists, but very happy scientists because they can do what they want to do. And the way these things normally work is when you engage with another discipline, the people there get to understand what AI might allow in terms of annotation data again. So after this, we went on to a second project, which is another thing they really want to do, is to annotate the behaviour of the chimpanzees, because they're interested in how behaviours were learnt in terms of the children from the parents. That's what they wanted to do. So their annotation task now is the behaviours. They're interested in nut cracking, eating and drumming actions of these chimpanzees. So I'll play you an example. You see this guy at the front, he's using stones. He's going to use the rocks there to break the nuts, and then he can eat them. Okay, they wanted to, again, go through these hundreds of hours of video and find these actions. Now, in normal computer vision, actually finding actions is quite a hard task. So for chimpanzees, it's a bit harder. But what we had here, you heard the sound there. So this is audio-visual. So the way we developed this was to use the sound of the nuts being broken as well as the action, and then we could label the data. So this is what we did. So you'll see example here of actions of nut cracking and the actions of eating. After they cut out the nuts, they eat them. And now some actions of drumming, which is what they do to signal. The drumming action. Here's a night camera. So again, we annotated all this data, and the same procedure happened again, that we then published a paper on the method so that others could do it. We released code so others can use it. And we're still collaborating with them, and this led to other collaborations. So these sort of engagements are really valuable for us and for them. So I'm going to sort of distill out the critical steps in this process. So the first thing before we can actually annotate the data is we have to get data from them. And actually this is a really difficult task because often in these disciplines, data's on various tapes in drawers, you have to get it all together. That takes a lot of time. Then they have to do some annotation themselves because, of course, we don't know the face identity of the chimpanzees. So they have to annotate a bit of data so we can train the methods and evaluate the methods. And in doing this, we get to a common understanding. We get to understand what they're really trying to do, and they get to understand a bit about the machine learning method of keeping training separate from test, just these basic ideas. Then once the data, we're ready to annotate the data, then we develop the models and run it across the data. And then we release code and annotated data. And then there's an initial paper which describes what we did, and the scientists themselves in their own discipline can do these follow-on papers where they make use of their own data. And usually then there's more engagement as we both realise what the potential is. So that's one example. I'm going to show you two more examples a bit more briefly. So the next example is in materials. We've heard about materials. Here, this is an analysis of X-ray videos of crystal formation. So it's very high temperature and the crystals are coming out of solution. This is for aluminium alloy. And you're going to see these are some frames from a video as these crystals come out. And the task that needed to be done here was, the annotation task was to detect these crystals and track them and ultimately count them. This was with materials at Oxford. So what you'll see here is, on the left you'll see a video of the crystals coming out, and here's the count that was computed on this curve. You'll see two curves. One of them is the ground truth that they annotated for the sequence, and the other is the prediction. Now, they follow closely together. But again, these people it's very easy for them to get data. They've got terabytes of data, but unusable without this type of annotation, which now they can do. So again, I've got a testament from the person who we did it with, who said, this was transformative for them because they couldn't use this data before. So again, same story. We published a paper on the method, and now that enabled this person to get a grant for further funding. So it really liberated them. Okay, last example is conservation. This is an example here. This is monitoring penguins in the Antarctic as a way of monitoring climate change. So this particular scientist has put many cameras around the Antarctic and collected data over multiple years. He has, at the time we did this, he had 500,000 images, many, many videos. And the task was to detect the penguins and then count them to see how that changed over the years. So for this, we built a method which could do this. You could plug in images or videos. And what you're seeing on the left are these coloured polygons are the nests. And what they wanted to do was to monitor how often the penguins stayed in the nest, so the average occupancy of these nests over time. And this is calculated, each colour corresponds to one of these nests, and this is over time in hours, which we could just leave running and running and running on the data. And actually, sometimes these problems are so challenging that we have to develop new methods. And so in this case, it's actually quite hard to distinguish penguins from a computer vision point of view. They look like stones. You have to detect them in all these different climate conditions. It can be sunny, it can be snowy. If they get smaller as they get further away because we're perspective, so it's quite a challenging task. So we had to actually develop a new method for this. So we published in our own computer vision literature a new method for counting. And then there were publications on the method, and the data was released, so others could use it. Okay, so summarising the message of the talk is that I've given you some examples of where in various disciplines the scientists had data but couldn't use it, and this is something that we should all be helping with. Once you engage in these projects, it opens up new research. The scientists look at the data and before they could see a few thousand images, but now they can see the whole thing. And so it lets them think of things that they wouldn't have thought of before. And then a couple of unexpected benefits is, one is they have to organise their data, and that's obviously a good thing because they have to get it all in one place. And also, somewhat unexpected, if we're going to annotate their data, they have to be very precise about how we're going to annotate, what the annotation scheme is going to be. So they have to actually not be vague, but be really clear on what is sort of levels and tags. And this previously might not have been done. Okay.
Thank you. Okay, according to the schedule, we're exactly on time. So are there questions?
Is it on? Yes. Related to the protein structure talks, I was intrigued by the fact that neither of you mentioned cryo-electron microscopy. And so I'm curious if you have any comments as to how that relates to what you've seen.
Oh, well, that's the limitation of 15 minutes.
Yeah.
I think it is. So for people who don't know, there's another method to determine protein structures. It's becoming increasingly common, something like a third of depositions now in PDB, something of that order, where you flash freeze individual proteins, you collect an enormous number of the worst images you've ever seen of anything, and you take 1 million of the worst images you've ever seen of anything and make sometimes a really incredibly high resolution image of the final product. And there's some lovely AI problems actually within that, like, you know, how do you do this reconstruction that people like Shor Shares, Ellen Zhang work on? But I think also it's interesting because it has changed the nature of what gets deposited in the PDB. So in between kind of the release of AlphaFold 2, and then we had basically retraining AlphaFold to a really AlphaFold multimer, which is a very similar system, on new data, and we saw qualitatively better performance on very large proteins because cryo-EM actually can't be used on small proteins, but works much better on big ones. And so we would see data shifts, and I think it's maybe one of the interesting points is, in machine learning, we always train our systems in the past and use them in the future, right? So I'm a big believer, you should always have a time split in your data. You should always be your test data should be newer than your training data and well separated. But one of the reasons is because what people are interested in, both in subjects and the methods they use, are constantly shifting. And cryo-EM is a really great example. There's some really exciting methods in whole pictures of cells at something like 15, 20 angstrom resolution, cryo-ET. I don't know, it's all wonderful. We should have a whole day on that. It's what we can do tomorrow.
Yeah, I don't have a lot to add. I think from a CAS point of view, it's really changed the nature of the targets we've got. Partly, people no longer send us simple proteins because they know things like AlphaFold will work. And so we get these big, hairy things with many components, a lot of mobility and so on, and often alternative conformations. So it opens up John's indicating a whole new set of challenges in structural biology.
Okay, I think it was really interesting to see among your talks how the algorithms that you're talking about seem to affect the economics of data. So, for example, with AlphaFolds, this seems like it could devalue the acquisition of new protein structures, right? If a student is going to spend 2 years to get a good structure of a protein, and it ends up being what AlphaFold predicted, it's like this is just a waste of time. So versus, I don't know, what Andrew and Carolyn were talking about was maybe more like the algorithms make each bit of data more valuable than they were before. So how do y'all think about this?
One thing we definitely thought a lot about on AlphaFold, especially as we're getting ready to release it, like, what is going to be the effect on that grad student that spent 2 years? And we could literally see, and you could see this on Twitter, someone said, they looked in the AlphaFold database, they said, how did they get my structure, right? And someone said, did you send it, did you send it by email, or like all these things. I think it affects science. Certainly every technological change affects science, affects what you can collect. The good news is very few biologists are structure-only structural biologists. There aren't many people whose only job it is to solve structures. They're solving structures as an inconvenience to go do that second thing that they really want to do, which is understand how this protein is associated with this disease. So in some ways it helps them skip. In a lot of ways you still want the experimental structure. Either they're more accurate, or they have a different error profile, or they include components that we can't predict yet. There are lots of reasons to want experimental structure. But at the same time, it is an opportunity, and some of the best science is really adapting and saying, well, what can I do with 100 million structures that I was never going to collect 1,000 structures for my study, but now I can ask about the evolution of this protein family across many, many examples. So we're seeing all sorts of things. But it does change. I mean, whenever there's a really, really powerful new technology, the right set of things to do in science will change.
Yeah, I think I know of cases where people have had their grants rejected because the reviewers, ignorantly or informed, say you shouldn't be bothering with this, you can already calculate this. But on the other hand, the AlphaFold technology and others have been incorporated into the main experimental pipelines in structural biology and actually speed up a lot of the work. So if you ask the experimentalists, I think they'd say it cuts both ways and changes things, as John says.
Yeah, I mean, production is still going up. I think it does affect the concept of, like, do we invest in a new beam line that will turn on in 10 years? And that becomes a harder question.
Also, I think, I mean, Pushmeet put it nicely this morning, right? I think you guys chose the problem very carefully, where there was one problem where there is a lot of data, it's well structured, and it can have a huge impact. Whereas I think the problem I talked about is actually a different problem where, the data is not there, so now AI can help us figure out which are the most valuable data sets that we should be collecting in order to then generalize more. And I think it's exciting to have these very different types of questions. And then, of course, we also need very different types of tools in both of them.
For my part, once you know that AI can take useless data and make it useful, then the scientists will go out and get a lot more data because it liberates them. We don't have to take this video and spend 10 years annotating it now. We can just get many, many videos and we can acquire a lot more data. So it's liberating in that respect.
I have a question for Dr. Phillips. You mentioned bias and your own work on bias, but I think that there's been recent work as well about racial bias and facial recognition and the consequences that this has on, especially I think in commercial systems, right? Like the ones that are deployed by police precincts and are actually used by active identification of people. And I'm just wondering, from a measurement perspective, given that NIST is really focused on measurement, do you actually make categories? Do you break down some different statistics? Because you were kind of plotting the average per 1,000 identification, right? But does that break down, and do you track that over time as well? And is there a similar evolution, or how has that changed?
So there are two sides to this coin. One is we do work with forensic examiners and other people who set policy. And we don't tell policymakers what to do, but we can tell them to give input for the numbers. So the cases of where people take low quality images and run them through, there are people who do examinations saying this is not the way it should be done, and there are recommendations going out. So I do know we interact, and I do know there's an active community doing this. There's a report that NIST put out in 2019 by my colleagues Patrick Rother, May An, and Kai Hoka, who they got 18 million face images from different sources. They ran, if I remember the numbers, 189 systems from 99 producers. It's about this thick, and it's very comprehensive. So you can go in and look at that about actually what the numbers are for different databases at different points and different decision levels. I believe they're now thinking about doing a new one to update it because it is now 5 years later. So these are active areas of investigation that, and NIST will put the numbers out to help do this. There's also another addressing the full issue with this, the National Academy of Science, Engineering, and Medicine had a study on face recognition and its implications, and that is now available from the website, who discuss both the technology and the policy issues associated with this.
Thank you.
I have a related question. So great presentation, first of all. So interested to know that you used an inherently social concept of race, which differs even between the US and UK in terms of its categorization. It'd be interested to know how did you define race in your studies, and did you apply it across different countries, or is it just US ethnicities within that?
So the studies we did here was data collected at the University of Notre Dame, who over a 10-year period collected a large amount of data, and we used self-declared race. So the person would call up and say, these are my race. It was follow procedure, but they're self-declared, and that in the social sciences is with my colleagues is the best standard for declaring race.
Yeah, this is for Jonathan too. So I was really interested in the very uniform improvement every year, and it would be understandable to me if like computer, programming ability or computational power kept getting monotonically better. That would be one thing, but you changed the databases that went into it. You took in, you started considering bias at different times, and yet it's still, kind of a uniform improvement. And you must have thought about that a lot, and just be interested in some further thoughts on that.
So we have studied bias, but that was separate than doing the improvement over time. So I first made a chart like that with that curve in 2010. I did another one in 2018, and the one 5 years later, and it's been consistent. The only thing I can think comes to mind is a function of both Moore's law, machines are getting more powerful at that rate. Also, when I did at the beginning, we did the ferret data collection, there weren't digital cameras. So we had a small number. And then we moved to these other data sets here, but also data became available on the web. You could just download because the digital camera was in download data. So it could be that some of the performers also downloaded data. By the way, this is a controversial issue, but I'm just saying what they've done. And I will say that all the data we use at NIST has been cleared by our IRB and our management. And NIST does not endorse any of these necessarily these other data sets. So it could be just increasing computational power and the availability to download it for people to download images or obtain images to train them is my most likely reason why you see the standard curve going down.
This is really a follow-up question to that, but it's for the whole panel. I also thought it was fascinating that you get this log linear improvement in quality. So my question for the panel is, do you see this in your domains or anticipate it in your domains, especially given John's explanation about why it might already be happening in his domain? And if you do, should we make some projections about the likely impact of this on the practice of science in your various domains?
I don't know, my experience with these kind of community challenge results and what happens is the really engaging thing is the surprise that I actually don't know what's going to happen next. I don't know. I didn't see deep learning coming, and so it's, well, it's maybe me, but there's been a whole series of those, and improvement has been a sort of punctuated equilibrium where things got stuck for several years and nothing happened. You think we've got to give up doing this. Nobody's going to do anything, and then suddenly, not only the deep learning things, but a new method comes out of the woodwork and really makes a difference. So, I guess my experience is much more nonlinear than yours.
So, I should emphasize that number is the best performance on a certain date. Mm-hmm. So it could be each time I have a dot, it could be a different performer. Mm-hmm. So that makes
the technology.
So it could be that.
No, that's for me too.
Pardon me?
The results I'm talking about are best results.
I mean, CASP was decidedly nonlinear. There was kind of a desert of CASP when people weren't getting better, and this I think you documented well.
Can't go to desert. But,
All right, not desert.
I think punctuated. There were certainly dull periods.
There were there were less exciting. I do think that. I think this is one of the most impressive examples for what you said on the dais. Not that we looked back and found out it was linear or exponential the whole time, but rather the claim that you looked in 2010. And actually, I will say you did have a little bit of curvature in there that was hidden under the line. If you had gone back and done the 2010 analysis, because I've noticed a lot of people say, look, it's been exponential forever. And then that statement is invalidated the next time because you have the multiple hypothesis problem that every time it didn't turn out to be linear over all the years, nobody trumpeted that one, right? So, but you said you tried this in 2010 or, that there was some predictivity? Because people have drawn a lot of these for general deep learning or improvement in perplexity and some other things.
So I think I know where the curve is, and that was at the beginning. And that was a retrospective implementation of eigenfaces. And we gave the I coordinates, so it wasn't fully automatic. So there's a caveat with that. But the other thing, going back to the quote, the periods, if you notice, I measured these on average about every 4 or 5 years apart. So it could have been, I just skipped over these, these points where, and that was more because at one point we did not do these every year, because there's a lot of effort. And about 2018, Patrick Rother, who now runs this, they went to ongoing. So algorithms can be, you can submit algorithms once every couple of months, couple times a year. It's a fixed number, but it's continually updated with the latest ones. So maybe if I took a finer grain, you'd see it flat for a year and went down.
Yeah, it could be.
So I think it's a really interesting question, and I think on the cell state transitions, we really don't even know yet what matters. So we have, everyone will give different numbers, but let's say we have like hundreds to 1,000 cell types, and we have all these different perturbations that we could do. We don't know yet whether it's more informative to do perturbations in many different systems or many more perturbations in one system. So those like very simple questions, I think we don't know. And that's really critical because what we don't know, what is the dimensionality of a cell, right? What is it actually that we need to perturb, et cetera, in order to be able to figure that one out? It's actually not even clear that the current models are really going to work so well in this system, because if we're thinking about cell types, it's actually very not continuous, the space. We should more think of it as a discrete space where we have these, you have this landscape, right? And you have these semi-stable states, and you will hardly never see cells in between them. So it seems very, very different than language. Or images, in fact, which seems much more continuous. And so it's not even, so even that is like the space that we're operating in is very, very different because it seems more discrete. So it's really, it might mean that we need many, many more perturbations. Even the protein space seems much more continuous when you're doing your perturbations than actually the cellular space. So I think there is a lot to be figured out.
So in machine learning, machine translation community, workshop on statistical machine translation has been running about now 2 decades or so. The annual shared task on news translation as well as the various other types of the, let's say, competition where the teams can participate. They're going to create a new test set every year using the news articles from the particular year only so that everything is fresh. And then there was a desert. And then that desert wasn't necessarily caused by the technological, let's say, plateau, but because at some point machine translation systems, let's say earlier, let's say 15 years ago, started to work well. And then all these companies actually stopped releasing their secret sauce. So in fact, most of the participants who are academic participants were not able to improve. However, when they checked the online systems, ABC, so those were, let's say, Google Translate and a few others, their scores were going up almost every year, monotonically. And then I have a question for John. Well, we have 3 Johns, but okay, generally. And in the context of, let's say, protein structure prediction here, so are we actually going into that kind of era where we're going to see that the systems are going to get better. However, these systems are going to be hidden behind the company's paywall or the company's, let's say, I don't know, like the technological wall, to the point that we'll see that it gets better, but not a lot of people actually get to participate in the innovation or using and then contributing to the improvement.
John's being put on the spot there, so let me say something.
Oh, good.
I think that there'll be some of that, but I think what happened with AlphaFold 2, there was initially a delay in release. It was well addressed, but it's not a criticism. But what happened was that people immediately began reproducing those methods. And so I don't think it's possible for anything to be under the hood for very long. I mean, I agree it'd be nice if the world were different. The world isn't different. I think there are commercial pressures one has to recognize and respect in some way.
I think the kind of other thing that I would say or think about as these is certainly there's various pressures or commercial pressures. I think there is a question, maybe an underlying question of how well academia is set up to do this kind of mid-scale type of work. So the AlphaFold team was around 15, you know, a fairly sizable amount of compute. The kinds of things that I think maybe the people aren't so large, but the compute becomes more expensive. I think that it is a challenge for the funding structures to make sure that the work can be done there, that these kinds of projects can, and academia has been able to produce some very large projects, right, CERN among them. But how do we think about this in the age of AI? How do we think about the ability to work on these kind of problems and also to deliver systems? And I think one of the other stories of AlphaFold is that it is, and what John measures in CASP are systems, right? And how do we think about system improvements? Also, how do we as an academic community, and I guess I'm on the industry side of this question, but let me say, how do we as an academic community also value performance and not just ideas we like for their aesthetics? How do we measure ideas by their percentage improvement in translation accuracy, not because you're excited to tell your friends about it? And I think one of the things that we did do really well within the AlphaFold work is be just as excited by these really kind of beautiful ideas that I like to put in my slides, and the other person who said, well, maybe that 8 should be a 12, right? And celebrating all those, because what we want to celebrate at the end are really great performance that enables transformative work in other people's science. And, you know, for them, it is a black box, and that's okay as long as they understand when it works and when it fails. But I think academia is not well set up to produce that, and especially kind of academic machine learning and NeurIPS style kind of papers.
Yeah, I think just to echo a little bit of that, I think that hopefully funding agencies are taking notice from a policy point of view. You know, one of the things people said, oh, it was sour grapes, right, when AlphaFold came out, is, well, they have all these resources and so on. But you can turn around and say, well, look at all these groups that took part in CASP working independently. I don't know exact numbers, but it's certainly comparable to what DeepMind put in, but it was splintered. And so what we should learn from that in terms of how to organize science is still working through.
So according to our schedule, it's now time for breakout groups. I don't know if we're going to get that nice...
Where to go?
I don't know what it is, assignment of people to tables.
On the back of the card.
Or if it's on the back of each of the placards denominating the table's number. Okay, I trust that we had excellent breakout sessions. I'd like to call on the rapporteurs to tell us about each of their tables. And this time I'm going to do it in reverse numerical order. I believe in countdown. So table 5, please.
Well, that was disruptive. I was thinking I have a lot of time. So on our, in our table, we talked about generalizability and how when these challenges can get motivate overtraining on challenge data set, and how the data set specific biases can enter. We don't know how to solve that problem, but it seems that somehow keeping the data fresh is really important because they kind of build in the bias. It seems to be a hard call somehow that has to be something that we have to solve going forward. Other questions that we talked about is how do you use the knowledge, scientific knowledge, and how do you generate new knowledge with use of AI? There was a question of like, somehow today we didn't hear a lot of the word knowledge, that specific vocabulary didn't seem to have been uttered too many times. That was an observation. And but the question is like, how do we generate new knowledge with AI? There is a level of how do we do difficult generalizations, and also is it possible to come up with something that's really truly disruptive and very outside of distribution. So these are the kind of things that we talked about. We don't have solutions, Questions, yes.
Okay, thank you. Table 4.
So we started by discussing some of the challenges that we are currently facing and how we see research moving more toward interdisciplinary teamwork and more focus on rethinking experiment design. So in general, the feeling of the table here was that we need a mix of domain area expertise and AI expertise in research. And then we moved to talk about the importance of trustworthy AI for science, and that in science, actually, there is usually a human in the loop, whereas in some of the use cases that were mentioned in the previous session, such as face detection, there were concerns, and therefore the question was, is the accuracy of AI algorithms better than the human error rate? And the fact that we need to decide whether to use AI based on the risk that we are willing to take. We then moved to talk about something completely different. But we did. And that was why actually Andrew Zisserman's talk has captured the attention of many of us. And that was because it was different to other talks in the fact that the annotation of data enabled further science, and the fact that it was not so much about quantitative results. We talked about how important it is to do research that goes beyond quantitative metrics. So, for example, finding or recommending a treatment for a patient. So something that is not necessarily a number. And the importance of learning what to measure. But that's not all of it, because it was reflected that some of the results that we have seen before have been important because the results were transferred also to other unrelated problems and were applied to other areas. And that's where their impact was important. And which raised questions such as, do we need optimality? Or how important is it to ask why when we are looking at the results of an AI-based study? And that also leads us to look at what happens when we have measurements and reporting, because it kind of focuses the attention. And while we have seen the good, we haven't talked about the bad as it directs further work around the same area. And the last thing that we discussed was about training. And to go back to our first discussion of interdisciplinary research that involves both domain area experts and AI experts. And we realized that even in a mixed team, there's a need for a common language. And I think that the most interesting insight here is if we're looking at undergrads today, then machine learning becomes a bit of the equivalent of what programming was a few years ago. So if you expect today that undergrads will already know programming, then today or in a few years from now, probably you'll expect undergrads also to know machine learning and then become experts in their science area. Thanks.
Thank you. Table 3.
So we had a varied and interesting discussion. One comment was zero-shot learning of foundation models and how unexpected it was when that appeared. And sort of led to another follow-on topic. One was the effect of fine-tuning, and particularly what could be the properties of the original model. When you change it and fine-tune it, you can change bias and other properties of that model itself. And one of the other probably more deeper questions was we've gone through a lot of number of examples and other stuff this morning about data and models, but there was a lack of discussion of foundational models associated with what we saw. And a comment was made that that companies can go out and raise money to produce foundational models, for example, cells, and what does it mean? Is it viable? So another thing we're talking about was how some new fields are resistant to deep learning. And the comment was that each number of fields, as it turns, have a deep learning moment when they have to accept that deep learning means a change in how they paradigms, and maybe need to switch to working working on new ways in their field. Another one was the statement that there seems to be a paradigm shift for evaluations coming on. So the ones, for example, we discussed there are static, and there's a need to move to variable ones. For example, looking at long tails, and the question is where you get long tails, and notice that scientific papers themselves are long tails because they're novel and published, but also the question comes, how many of the results in these papers are actually accurate? And finally came discussion about schools and our hiring lots of professors in AI, and what that means going down is that they'll train a lot of students, and how will this affect the community of large growing. Also, if lots of universities are hiring new professors, how are they going to get funding for grants or contracts when there's a large supply of new professors who need to bring in money to justify their hiring for new positions? And that about covers it for this table.
Thank you. Table 2.
So we had an interesting discussion. We spent a long time in understanding what are the challenges for AI, of AI for science. So we focused on 3, I would say, key aspects. One, we discussed for quite a long time on causality. So on one end, the need of causality was for science was deemed important. On the other hand, understanding when and why it is required remains a complex issue. And there's also issues of multiplicity in causal discovery, and there's ongoing debate on the necessity and on how to measure the causality, given that current models do not account for it. And we discussed training limitations, as AI models like chatbots are predominantly trained on observational data and not causal data. And this may lead to a lack to correctly account for causal inference. And then we discuss about other challenges. So we often want to tackle problems that act in complex systems that are presented in multiple stages, often uncoupled, and they need synergistic utilities, but oftentimes there is a disconnect between the objective of each of those systems. So we're thinking at systems where you have a prediction, and then this prediction will inform some decision. And so what should be here the objective of those AI systems rather than those AI tools? We need objectives that optimize directly for these downstream decision processes, and how do we do that? We also talked briefly about issues of discontinuity, which was risen in the last session. So that there is a reliance in modern AI systems on gradient-based models that often are challenging because they are challenged by discrete problems, by tackling discrete problems. And we discussed also about what are the characteristics for AI to make an impact in science. So this was also mentioned in the previous session. I think we need good data, a well-defined prediction challenge, and precise mathematical formalization of the problem. but when and where we can find this combination of those tools. We've seen great examples in the previous session, but there was ongoing debate on which one are other areas that are fertile over which those three characteristics arise. And we talk about evaluation and benchmarks. So there is a need to develop robust evaluation metrics and benchmarks that are specifically tailored to the complexity that are introduced by AI systems. And I think that covers it.
Thank you. Table 1.
Great, thank you. So most of our conversation was around the sort of intersection between universities and businesses in terms of AI. There were a few other things we talked about, but that was probably the main thrust of what we talked about. So we talked about, first of all, about data sets, access to data sets, in particular that there are a lot of data sets out there which are essentially funded by research councils, which are essentially funded by taxpayers. So what do we do if there's a situation where you have a big tech company who trains an AI model on a data set which is essentially funded by the taxpayer, but then that AI system is then proprietary to that company? So how do we ensure that not just academics, but also taxpayers are getting sort of good value for the sort of data that they're creating, which is powering these AI systems? We then talked a little bit about the sort of open source coding structure and how what's been learned from open source programming could be used to create sort of open source data sets and the sort of licensing that might be needed for data sets going forward. We then talked a little bit about sort of what's the sort of special aspect of universities which perhaps industry doesn't have, and we discussed how universities sort of have people. So the people pipeline, the supply of people is an important part of what universities can offer AI, and that kind of led into a conversation about there are some sectors, not AI, but other scientific disciplines where the industrial sector is already quite far ahead of universities. And could this happen in the AI space, where some companies will be so far ahead of universities that there'll be quite a big gap actually in terms of the quality of AI research in industry compared to AI research in industry? And then that sort of led on to a conversation around, slightly conspiratorially, around trade secrets and what the companies are not telling us. So they might release the weights of their models, but they won't necessarily say how they trained those models. And so is there a bit of a concern there that the companies are releasing some things, but they're not sort of telling us everything, and that maybe, actually I won't go into the conspiratorial part actually, we can talk about that over wine afterwards. Then we talked a little bit about the sort of the changes to the job market we're starting to see now, that a few years ago students were being poached even before they finished their PhDs to come and go and work at one of these sort of top companies for high salaries. But now, as more people are trained and have PhDs in AI and ML, it's perhaps now the case that students actually need to apply for jobs and actually need to do interviews. But that also, the growth, not just at the PhD level, but also the MSc level, means that there are more and more people who are going to be trained in this area. How is that going to impact the job market? But then how does that also impact students who working in other scientific disciplines? So if you're a biologist, so how much AI do you need to know to be a biologist now, or to be a chemist or a physicist? And so what sort of AI skills do those students and academics need in other disciplines? And then sort of coming towards the end, we sort of talked a little bit about our own experience of how we used AI to improve our productivity, talking about ways in which we've used AI to improve our sort of coding or writing skills. And I think the sort of general feeling was that the technology is very, very good, but it's not perfect. It's great as a kind of first draft. It also can be useful to sort of help you write a book review, but you wouldn't just submit that. You'd still have to sort of like go back and polish it. So it's a good start to improving our productivity, but it's not going to get rid of us just yet. That was the end.
We're within a few % of on time. And I think there's supposed to be drinks coming. Do we have any announcements that need to be made?
Yeah, thank you very much. And this is the point at which we thank our live stream viewers for joining us. and we're saying bye to them until tomorrow morning. The broadcast is going to resume at 9 a.m. British time tomorrow. So thank you to everyone who joined us online, and many thanks to all of today's speakers. Let's give them a round of applause. Now, logistical announcements for those present in the room. We're going to have a drinks reception and a dinner. It's going to take place downstairs, so if you just keep walking that way, you'll arrive to that area of the building. You are asked to please take all of your personal belongings, and you can store them by the coat rack just outside this door, because people need to clean out this room and make it ready for tomorrow. Now, before the drinks start flowing, some announcements about tomorrow. The bus is at 8:15 from the hotel tomorrow, and I promise everyone a better bus journey than this morning, because it cannot statistically get any worse. So bus is at 8:15, 8:15, and if you're joining us from somewhere else, please be here at 8:30 a.m. tomorrow. And a little announcement also about tonight. There is no bus after the dinner tonight. So you're free to walk to the hotel. It's a very pleasant walk, only about 20 minutes. The weather is nice and it's very, very safe, central London. Or you're welcome to continue your evening somewhere else. We are in central London after all, so enjoy yourselves. Thank you.